So thank you so much, first of all, for the invitation. It's a pleasure to be here, and also for the kind introduction. Can you see my presentation on the screen?
Yes, thank you very much.
Okay, perfect. So I go directly into medias res. I'm zlem. I'm a physician by training, an immunologist out of passion and out of desperation. I became a founder and an entrepreneur, and that is the story I would like to share with you today. And it actually is not only my story. Can I direct my slides there? Yes, I can. It's not only my story. It's a story which I share with actually a tribe of mentors and comrades. And two of those mentors and comrades are shown here, Ugo Sahin and Christoph Huber. One of them I married, the other one became like a father substitute for me, and they are not the only ones. We have been joined by many over the last decades on our path to turn our science as immunologists and physician scientists into survival, which was our vision from the very beginning. And this vision was born out of observations we made as physicians on oncology wards. We had to learn in the early 1990s, when we were working as physicians, that standard of care has not much to deliver for cancer patients. And there were so many times we were in the situation to tell our patients that there was no medicine, no standard of care against their disease left. So this was one experience. And in parallel, as scientists, we made the experience what science and technology could in principle offer. And the science we were in particularly interested in was immunology, specifically understanding that the immune system has an entire armamentarium of effectors which one could mimic or leverage or harness against cancer, inspired us. And our vision was to use the foreignness of cancer cells and to leverage and harness the immune system to achieve tumor rejection. And the strategy we wanted to follow as scientists and immunologists was that we wanted to identify targets on cancer cells which would allow the pursuit of specificity, the pursuit of distinguishing cancer cells from healthy cells. And we wanted to use these targets in order to develop and use potent new immune therapy platforms. And what also was key in our strategies was that we wanted to personalize medicines. And this was the vision, the scientific vision, and we worked hard. And as academicians at the university hospitals we worked at, we developed a plentitude of technologies and scientific concepts based on these three pillars and principles, which we thought could help to develop better treatments against desperate diseases such as cancer. And this happened first at the Mainz University Hospital, meaning in an academic environment. However, we understood at some point that developing new therapies in an academic environment would not be a sustainable and robust way to bring new medicines to patients. And first, we try to convince pharma companies to patent our early stage developments and innovations in order to do the drug or immune therapy development. But we very fast understood that for pharma companies, our innovations were simply too early and not validated enough. And so the only path forward which we saw was founding a company, our first company, Ganymede, which we founded in 2001. And this shiny building was actually not how it started. It started much smaller, not exactly in a garage, but in a very small laboratory room at the university hospital, with an activity which first was about figuring out how to set up an enterprise, a biotech company, and how to finance it. And scientists, which we were, we started to read literature, which would help us to find solutions to what we wanted to achieve. And because there were so many challenges, we were not prepared for IP strategies. How should we deal with that? Finding the funding and investors, we very soon found out that we would need venture capital, which was new to us with all the rules it came with. Then managerial skills were needed, which we did not really were trained for. We needed to think of business models and drug development concepts, processes, which we were not used to. So these were many new challenges for us. And what we found out was that one important lesson learned in this process was that you can't do things half. Yeah, you cannot announce that you want to develop your product candidates in a biotech company and then find people who do this for you. You have to do it yourself, and you have to do it with full power ahead. So there is no do or do not. There is no try. You have to do it with skin in your game. And what we also learned that I have not the training, I have never done this before, is not really an excuse. However, what you can rely on is that if you have the will to achieve something because you are inspired by your science and want to translate it really in survival and into something meaningful, then your will help you in this. So what we, however, also found out very fast was that there are rules in the venture capital business, and our dream to bring all the plentitude of things we had developed, all those innovations into a company did not work out because venture capitalists wanted to hear a clear and simple story, a narrative, and a clear path forward of a product candidate, which according to a clearly defined development concept could go into something which would at the end of the day become a product. So we had to pick the most advanced concept out of our armamentarium in order to build a company upon that. And the concept we chose were immune effector mobilizing antibodies. And this was based on which in 2001 Ganymede was founded as what at that time was the paradigm for biotech companies a fast, focused, and lean business model where it was very clear with what we would start and how the path forward would be. The modality we chose, monoclonal antibodies, was an established one. There were already other monoclonal antibodies. It was a validated drug format. However, what we wanted to do different was that we wanted to pursue specificity. The monoclonal antibodies, which were used at that time and were approved at that time, were directed against growth factors, which played an important role in tumor cells, however, were also present on normal cells, as shown here, the conventional ones on normal cells and tissues, also on tumors. And we wanted to target molecules or features of tumor cells which were really cancer cell specific and not expressed on normal cells, in order to direct the immune system against those cancer cells. And this piece of the model was new. And so we started, and I have to admit, we started too early. At the time we initiated our spin-off. We only were on the level of the concept and had our first target molecules defined and patented, but we even did not have the lead candidate antibody or IND supporting preclinical package. So that there was quite some path ahead. And that meant that in the years in front of us, from 21 to 28, we generated quite some progress and developed an antibody and reached the portals to clinical entry. But at that time, when we were ready for going into the clinical stage, our venture capital investors, and we had at that time point more than 30 shareholders in our small company, were at a stage where they needed an exit. So they could not and did not want to finance the company anymore. And in such a situation, for a biotech company which has not already reached clinical stage, there are only two sort of paths. One path is to go into insolvency, and this has happened to many, many companies which started with us, or find someone who would acquire you, which is difficult if you only have a preclinical product. And we had this problem. There were two potential solutions which were not attractive for us, and therefore we, in this desperate situation, decided to look for investors who would share our vision, which was very different from conventional venture capital. And we tried hard and were able to find such investors, the Strömmann brothers, which were much more driven from the vision to develop an original product, an original medicine against cancer, rather than being interested in a typical venture capital model with an earnings exit. So their interest in our company and the fact that they provided our original investors an exit and then committed themselves to supporting our clinical program was rewarded. A couple of years later, we were able to show in a proof of concept phase 2 clinical trial in advanced gastric cancer, unprecedented data. We could show that our antibody, which was first in class modality, was capable of near doubling the median overall survival of patients with gastric cancer. And this then initiated a beauty contest of interested pharma companies who wanted not only to acquire this product candidate, but also wanted to acquire the entire pipeline of monoclonal antibodies we were developing. And so that we were able to sell our company with a high potential product with proof of concept in phase 2. And after doing that, the question was how to continue. In parallel to those last years in Ganymede, we had been busy. What we had done was that we had in parallel generated a public-private ecosystem. Not only Ganymede had been founded, we also had founded an organization, the KIMT organization, which was founded by Christoph Huber, which in the meantime is the largest European cancer immune therapy organization, which in the last years, in all those years, developed to thought leading association and organizes every year annual meetings in which we bring all stakeholders of immune therapy development together. In addition, we learned that it's not only academic institutions and universities and companies which are needed in order to translate science into survival. You need other types of organizations as well. And we founded a translational institute, the TRON, in which we basically do the part which cannot be delivered by an academic institution and also not by spin-off or a company. And we also mobilized our entire region by founding a cluster for individualized immunotherapy, where we ensured that cooperations and partnerships financed by grants between academic institutions, universities, and companies could be built. And one more thing, we also founded our second company, BioNTech, in 2009, because there were all, as you remember, all the other arrows in our quiver, which at that point were developed to a stage where they were of interest for being pursued in a drug development company context. And this model was an entirely different one, not the conventional, fast-focused, lean biotech model. We choose to build a horizontally and vertically integrated company in which we organized all our disruptive platforms in individual business units. And one of these platforms was particularly close to our hearts, namely cancer vaccines, which is one of the ways to harness the immune system. And the dream we had was to use cancer vaccines for personalized cancer treatments. An ideal vaccine, we thought, must first of all be tremendously potent, because by the time cancer is discovered, cancer has already invaded the body with billions and billions of cancer cells. And if you want to defeat this disease, it becomes a numbers game, and it requires the deployment of a strong immune response. And there was another additional feature which we wanted to have, as I said, in our cancer vaccines, namely that they should be truly individualized. And the reason for this is that the root cause why current cancer therapies fail is that each patient has a unique cancer, and then one would really want to tailor vaccines to the genetic profile of each individual cancer and to produce them on demand and customized for each and every patient. And this was our dream, which sounded science fictionally, but as Ugo Sahin, one of my comrades, always says, just pretend we don't know that this is possible, and let's just do it. And this was exactly what we did. We tried different platforms and found that mRNA, as the most primal information technology created by nature, could be the ideal platform for developing cancer vaccines. Back at that time, mRNA had a fundamental problem. It was of low potency. Even high doses of mRNA produced little protein and consequently also little effect. And this was clear to us from the very beginning, and we invested many years in order to optimize this platform and make it an optimal vaccine platform. And on the next slides, I have three of our core innovations, three of many. The first innovation is about molecular engineering and design to increase mRNA potency. The goal of a vaccine is to make your body aware of the identity of the enemy. And mRNA does this by making your body print the wanted poster, meaning manufacture the protein that is the molecular feature of the enemy. By nature, mRNA not only delivers the blueprint of that protein, the wanted poster, for the body's cells to produce, it also contains a set of instructions and how to produce this wanted poster. And our team spent years researching each of these elements to discover designs that would help significantly increase potency. And this resulted in a more than 1,000-fold increase in the vaccine's efficacy to trigger the immune response and the immune system. The second innovation was about the body hack to deliver mRNA to the right cells, the cells that matter. The immune system has specialized cells called dendritic cells, which naturally act as high-performance trainers. Once they identify the enemy, they are able to activate the various forces of the immune system that are gathered around them. Dendritic cells are not all equal. The most capable among them are found in the so-called lymphoid organs, the body's network of immune system boot camps. And ideally, a vaccine should deliver the wanted poster to as many of these cells in as many of these lymphoid organs as possible to train their troops. And we again spent years in order to develop liposomal wrappers, which once administered would transport the mRNA exactly to these cells and ensure that the right troops would be trained. And the third innovation finally was about rapid manufacturing on demand. Our efforts to develop individualized and personalized vaccines have not only made us obsessed with high potency, which we addressed with the first two key innovations, they've also trained our senses of urgency. And the process from genome analysis of a patient's tumor computer-aided design of a vaccine of unique composition, and on-demand manufacturing of this customized mRNA vaccine ready to administer. This process has been a race every time against the patient's growing tumor. And we started 2012 to 2014 with optimizing this process. At that time, it took us 3 to 5 months for each patient to manufacture their individual vaccine. In 2019, we had it down to 3 to 5 weeks. And with this vaccine, as shown for this clinical trial here, we indeed managed to get patients' recurrences here, as an example, in metastatic melanoma down significantly, and could thereby also address our vision to build better medicines. Which means that on that fateful weekend in January 2020, when we learned that we actually were already in the midst of the pandemic, we were able to build on all those years of perseverance and grit. We had developed the mRNA technology, and we had full command over it, and our teams were trained to go quickly through the process from genetic sequence to vaccine design to manufacturing. And this was the reason why at that point we could pivot our company and generate a COVID-19 vaccine within light speed time, within 10 months, but following scientific, regulatory, and ethical standards. So what we learned, not only in those last 1.5 years of developing the COVID-19 vaccine, but all those years in building an ecosystem of companies, but also public institutions, is that, yes, you can turn something like science, which seems small and powerless, into survival. And what we think you need for this is that you need to let your vision be your Northern Star. Founding a company is a component of the project plan on your path to this vision. We also learned that it pays off to build a tribe of mentors and comrades, which will support this path, and also to forge partnerships based on trust. We also learned that it's important to take over radical ownership and accountability, to leave your comfort zone, to put skin into this game and bring forward your company yourself. We also learned that courage and humility are a good modus operandi. You need the courage in order to make all those bold decisions, to enter uncharted territory, but also humility to learn in that process. And an important last advice is follow the science. Thank you very much for your interest.
Thank you. Thank you very much. Also, thank you, dear audience, for the first questions that we got. And let's start by reading the question from Roland Meyer. How have you developed your relationship with the university? What learnings do you wish to share for how universities can help spin-offs?
It was basically a mutual education process through which we went with the university, specifically the people, and people matter, which were on the university side. And this learning process was very much oriented in the joint understanding that we wanted to create something which had to be in a public-private partnership, and we defined the challenges we had on this path and developed solutions together. And I think this approach where there is not a clearly defined technology transfer or development plan, but rather solutions are found based on the specific challenges a spin-off has, is a robust way to deal with the still present challenge of universities supporting their spin-offs in a meaningful manner.
Great. I'd just like to read one comment, which also includes, I think, our opinion in studio. We were talking while you were giving us this keynote. Fantastic and inspirational talk. So thank you also on the behalf of the community. Second question from David Hoy. What was your experience with universities' intellectual property rights? How to get a patent out of your academic research that most likely has been published already?
I didn't get that question. What is the research is published?
So the question is, what was your experience with universities' intellectual property rights? And then the concrete question is, how to get a patent out of your academic research that most likely has been published already?
Yes, first the last question. That's certainly an important point that you should also, as an academic scientist, not publish before you patent, and we adhered to that concept. So nothing we patented was published before patenting. That's very important, because this is what venture capitalists look into when you want to spin off, and that's indeed frequently a problem in spin-off negotiations. Our university had a very straightforward approach of providing us with patents which we had produced or generated at the university, and not asking for direct payoff, but making or asking for royalties in the case that we would generate value out of these patents. And this was a straightforward and very pragmatic way, which was very important in the entire concept.
Mm-hmm. Also, one question from my side. I can remember that Christoph Huber told us in Innsbruck that you founded a non-for-profit company called Thron, and he said that this would be a good best practice examples for universities worldwide. Can you explain a little bit how Thron works?
Yes, Thron. So we made a lot of experiences when we founded our first company, and one of those experiences was that you need certain skills and expertises which you don't have at an academic group. For example, a deeper understanding of patenting strategies, a deeper understanding how to design the content of patents, but also a deeper understanding how you design your scientific project such that development relevant questions are tackled, and the questions you tackle are not only purely academic. And those concepts were formalized in Tron, so that the way science is done at Tron is directed towards preparing indeed drug development processes, IP-wise, but also content-wise and data package-wise, more aligned to what a company would need.
Great. Maybe a final question from Philip. Amazing talk. Thank you for your insights. Obviously, if anything is confidential, you don't have to answer the question. But he's asking, what are your main learnings from developing the COVID-19 vaccine? Will this help to bring personalized cancer vaccines on the market sooner than maybe expected?
Yes, as I pointed out, we have learned from the personalized cancer vaccines, technology-wise and manufacturing-wise, in order to be able to develop the COVID-19 vaccine so fast. And in the context of developing the COVID-19 vaccine, we had to further refine, for example, manufacturing processes, had to upscale manufacturing processes, work out logistics, but also improve our platform, our mRNA platform, even more towards potency. We had to learn about regulatory processes. We had to educate regulators all around the world about specifics of cancer, of mRNA-based vaccines, about the platform, and all this will now feed back into our individualized cancer vaccine development and will accelerate it. This is the expectation.
Perfect. Maybe just really the final one from Christina. Thank you for this inspiring and amazing keynote. How did you manage to keep going, even when all the financial odds were against you? How did you find the strength? I think this is an important question for many researchers.
Yes, those phases in the life cycle of a biotech company are the most difficult ones. When you reach the end of your financing round and have to refinance. And the only way is really to get motivated by your science, by the data you have, and by the understanding what meaningful difference this data can make, and to invest all your energy to get that financed.
Perfect. Thank you very much. I would like to close by quoting Clara Brandstätter. Really powerful keynote. Thanks. What a start into today's conference. Thank you very much, dear zlem, and see you soon. All the best for all your further projects and ambitions and whatever you're up to.
Thank you.